So today in lab, or well, next week, usually it would be today, sorry I get so used to saying things, the scientific terminology that we're going to use in this lab are things that we're going to use throughout lab this entire semester. And you should know what these three things mean. You should know, first of all, what the control is. So the control is something that is constant between all of the experimental groups. And then independent variables are the variables that we're changing between experimental groups. And the dependent variables are the things that we measure. So as we are doing this experiment this week, next week. As we're doing this experiment next week, we will be keeping one variable constant so that we know that that is not changing, and then we'll be changing little things like changing temperature, changing the pH, changing the type of tissue. And as we change those things, we're going to measure to see what is happening in this experiment. So in this case, we're measuring the amount of bubbling that we see in this experiment. And so after lab, you're going to be asked to answer some questions like, what is causing the bubbling to occur? Or why did some substances bubble more than others? And we're going to be using a scientific metric of comparison. One thing that's important for scientists to be able to do is to quantify the amount of things that we are measuring in the experiment, so that if scientists in the future want to do this experiment themselves, that they can say, you know, oh, I also got thousands of bubbles, or I also got too many to count. It's important to be able to have that metric of comparison. Otherwise, what one scientist says is a lot of bubbles may be completely different than what another scientist says is a lot of bubbles. So we try to make things as accurate as possible so that we know we're talking the same language. We're doing the same experiment. And so here we are now at the end here. I escape out of that, close out of that one, and let me look back and see how we are doing for time. Okay, 24 minutes sounds about appropriate. So let's go on, and we will do the PowerPoint from Lab 5. I think that when I do this video, I'm going to do a little bit of video editing when I'm done. I'll probably split it up into a couple of short videos so that if you want to watch them, like 4 little short 10-minute videos, or you can watch them all together. It's totally up to you. But let's go through and talk about DNA extraction from cells. So you can start to see some of the major concepts that we're talking about here in biology. Talk a lot about homeostasis, about things maintaining equilibrium or being balanced. Once things get out of balance in nature, that's when stuff starts to break apart. We talk a lot about controls, making sure that we are keeping things constant in experiments, so that, because if you change a whole bunch of variables, you never know which variable it was dependent upon. So the control is the thing we always keep the same. So we have then our independent variable, the thing that we change every time, and the dependent variable, the thing that we measure. So we'll see that in this experiment too, and I will point that out for you as we go along. Get some more of my coffee. Maybe I start talking faster as I go along with this coffee. Okay, skip the text message for right now. Alrighty, let's make that full screen.